Alright, welcome back. In this video, we're going to go over enzymes. This is going to be a really basic overview of enzymes. In biochemistry, enzymes are everything. So here, going forward, enzymes is going to be your world. We're going to be dealing with substrates, enzymes, and products. And that's going to be literally the rest of biochemistry. So I think it's fitting we start out with the basics. So enzymes are proteins that are mostly globular. So it's like, they look like a globe, a sphere, for example. And what they do is they help speed up chemical reactions in the body. Enzymes are known as catalysts because what they do is they speed up reactions, but the enzyme itself is not degraded in any way. It's recycled in your body. So here's what a typical enzyme looks like. It does kind of look like a sphere. But what enzymes have is something called an active site. There could be many active sites. I drew two here. What active sites are is a special area in that enzyme where the reaction actually occurs. So what do you mean by reaction? Well, what's going to happen is we have a substrate. So think about a substrate as any molecule. And what a substrate is going to do, it's going to bind onto an enzyme, onto the active site. And what the enzyme is going to do is on that substrate, it's literally going to strain that substrate. It's going to basically strangle it. I'm not even joking. That's actually how it works. It strangles that substrate. It strangles that molecule. And it helps break the bonds in the molecule. And it also brings molecules closer together. So areas of the molecules come closer together and they react. So our goal is we start out with a substrate and we're trying to get to a product, a brand new product, a brand new molecule. So for example, let's just call this A, and then we call this B. So our goal is that we need to have a substrate, so we have a molecule, which is A, and we need to get to point B, which is the product. So what we do is that this is called an energy versus reaction path graph. What this is showing is how much energy is actually needed to get to the product. So everything in our body requires some type of energy. Nothing's free. So what we need to do is we start at a substrate, and notice that we have basically this line here. We have this uphill. So the energy is increasing as we go in, as we go further. And then we dip down when we get to the product side. So we require energy to do so. But notice something. It takes a lot of energy to get from here to here. So much energy. This is wasteful for our body. Our body could be using this energy for other things. So this is where enzymes come into play. What enzymes do is they lower the activation energy. So this hill we described here, from the baseline to the top, is called the activation energy. This is the amount of energy that's required to get to the product side. Well, what enzymes do is they basically cut that activation energy. We're reducing that activation energy. So this is the red line, this red dotted line now. So notice this red dotted line. So we went from originally here to now here. So we cut the activation energy in almost in half. So it's easier for us to get to the product side so we can make more product. So that's literally how enzymes kind of work. Now there's something called the transition state. This is really important. The transition state is the top of the peak of the activation energy. What this is it's saying that once we're here, we could go either of two ways. We can actually go to the product side and make our product, or we can actually just go backwards and go back to the substrate, where we started from. Sometimes enzymes are designed so they're not that efficient. Sometimes we can make so much product. Sometimes you make so little product. This is called enzyme turnover rate. So let's look at this graph here. Let's look at carbonic anhydrase with a turnover number of 1 million. Lysozyme has a 0.5 turnover rate. What this means is that per minute, we can use carbonic anhydrase and make 1 million products per enzyme unit. As lysozyme, we only get 0.5 per minute. That's really low. So the point I'm trying to make here is when we get to this point, it's not a guaranteed 100% we're going to make products. We could actually roll backwards and go back to the substrate. That's the point I'm trying to make with the transition state. So, enzymes end in the suffix ase. So let's go over some examples. Ligases, so we have that ase, join molecules together. It's kind of like super glue, or bonding things together, molecules together. Amylase breaks down carbohydrates. Amylase is located in your saliva. So when you're actually chewing on food, you're actually breaking it down with, of course, your teeth, but also amylase. And I can prove it to you. If you have rice, say you're eating rice, next time you eat rice, you should do this, is put it in your mouth and chew it. And it's going to taste just like rice, but chew it a lot. Keep chewing, don't swallow, just keep chewing. And over time, it's going to taste sweet. It's going to taste like sugar. The reason is because the amylase actually broke down that carbohydrates into single units of sugar. That's why it tastes like that. So this part breaks down carbs. Lipases break down fats, and proteases break down proteins. DNA polymerase, another enzyme, synthesizes DNA molecules. Oxidoreductase is another enzyme that transfers electrons to other molecules. Enzymes can do so many different things. And we're actually going to go over many of the functions when we go over the different pathways like glycolysis, gluconeogenesis, the Krebs cycle, the Cori cycle, all that kind of stuff. So now let's talk about enzyme denaturing. So enzymes are proteins, and proteins can denature. What denature means is, remember in the last video, if you watched that, please watch that. But if you did watch it, or you just know this background knowledge, proteins need a fold to become functional. So we had that primary structure, we had the secondary structure, we had the tertiary structure, and we had the quaternary structure. And the quaternary structure is the functional protein. Well, we actually can go down that levels list. So we can actually go back to a tertiary level, back to a secondary level by denaturing. What we're doing is basically making that protein or enzyme useless. It doesn't do anything. So that active site we talked about here gets destroyed. So it can't do anything. No substrates can bind. So methods on how to denature. There's many methods to do it, but I'm just talking about like the body specific ones. So temperature. Heat can denature proteins and enzymes. Cold cannot actually. So lower the temperature, it really doesn't do anything. But higher the temperature, it actually can denature. I'll give you a really good example. Fevers. When you're sick, so you have an infection or a viral infection or whatnot, you get a fever. A fever is actually a really good thing because it's your body's defense mechanism to denature the proteins and bacteria and viruses. Because remember, bacteria and viruses do have proteins in them. They have DNA. So what they're doing, what your body's trying to do, your body's really smart and it knows how to kill infections and viruses. So what it does, it raises its own body temperature, which is a fever, and it tries to denature the proteins in bacteria and viruses to kill them. Now, there's a bad side to it. If your fever gets too high, like 105 degrees, that's like dangerous, you should go to the ER. And the reason is because you can denature your own proteins at that point. You have proteins in your body, like your brain. Imagine denaturing the proteins in your brain. You can go brain damaged. You could literally die from that. So that's how you can die from fevers. So it's not necessarily a good thing to always, immediately take some Tylenol or ibuprofen when you have a fever. No, not really. It's because you should let that fever continue so you can actually kill that bacteria, kill that virus. Now, if it's 105 degrees or 104 degrees, yeah, please take something. Don't go on because it's dangerous. But like 102 fever, in 101 fever, it's actually good because you're trying to denature the proteins. Your body's trying to denature the proteins and bacteria and viruses. pH. Different pHs can actually affect enzymes. Here's another example. Enzymes produce in the mouth. Let's actually do amylase. Amylase is produced in the mouth in saliva. When it enters your stomach, which is a really acidic environment, that enzymes, the amylase, actually denatures and becomes nothing. It doesn't do anything. So it's actually responsive in pH. Chemicals. All right. This is why you shouldn't drink bleach. Because you will denature your proteins and you'll die. Besides the liver toxicity you would get from drinking bleach, you'll denature your proteins and enzymes and you would die. Your body relies on enzymes to do processes. There's so many reactions happening in your body, millions of reactions. And they require enzymes, otherwise it'll take too long for things to happen. You would create stuff that'll take way too long and you would die. Please don't drink chemicals. Don't drink battery acid or bleach or anything like that. Don't go to the grocery store, get Clorox bleach and drink it. Please don't do that. I beg you, don't. But yeah, I think it's kind of self-explanatory. I hope it is. I don't know my audience, but come on. You're in biochemistry. So don't drink bleach or battery acid or anything like that. Just don't. Now, that's the overview for enzymes. The next video I'm going to do is about the chymotrypsin mechanism. We do mechanisms in biochemistry. This is the biggest one you basically need to memorize. I honestly don't know why they teach this specific mechanism in biochemistry. There's so many of them. I don't know why they choose chymotrypsin to teach. I'm assuming most of your professors probably going to teach you chymotrypsin mechanism. So we're going to go over it step by step. And then I think after that will be Michaelis-Menten equation. I got to check, but I believe it is. So until next time, later.